Okay, so the project that we've been working on in the lab relates to the spike protein that's on the surface. Probably all of you have seen pictures of what the virus looks like. This is a computer model that we generated in our lab. Mostly we've been working at a smaller scale than this, but trying to work up to having a full viral envelope model. And so I want to talk a little bit about what the spike does for those of you that don't know how the virus works. The virus needs to get its RNA into the host cells and through the membrane, and it does that with the spike viral fusion protein. And so down in the bottom here, I've got kind of a cartoon, and going from left to the right is the process of viral entry. This is the viral membrane on the bottom, and here's our spike. It's a trimer. It's got two subunits on it. One of them is this orange one, that's the receptor binding subunit. And then the blue one is the subunit that's responsible for membrane fusion. So there's some motion in this that I'll talk about. Then it's going to interact with a host cell receptor and SARS-CoV-2, that's ACE2. Then after that, these S1 subunits are shed, leaving behind just the S2. It undergoes a conformational change that attaches itself to the host cell, and then another conformational change that brings those membranes together and leads to membrane fusion. And so this is common, not just to SARS-CoV-2, but this is coronaviruses, and many viruses have these viral fusion proteins. And so the key event really here is that interaction with the ACE2 and all of these conformational changes. Because this spike is so important, it's the target of your immune system. This spike is most of the vaccines that are in development. They're either spike proteins, RNA that codes for the spike protein, things like that. And the antibodies bind to the spike on this flexible domain up on the top, this orange receptor binding domain, the RBD, I'm going to refer to it as. That red domain is not very well conserved, and so antibodies between different coronaviruses are not transferable. There aren't broadly neutralizing ones because the sequence is so variable. The sequence on the fusion part, the blue part on the bottom, is very highly conserved. And so this is our cartoon here, but really we don't know a lot about the structure and how this works. We know from cryo-EM what these look like in the pre-fusion state, and we know what it looks like in the post-fusion state, but everything that happens in between is pretty much missing, or we have little bits or fragments of things. We don't have structures. So that's really what we're trying to figure out, is how this works and if there are opportunities to interfere with it, particularly ones that would not involve vaccines, which are our most likely outcome here for SARS-CoV-2. But we need to be prepared for future coronaviruses, and this S2 is probably a better target for something that's broadly neutralizing. So this is what the spike looks like in cryo-EM. Cryo-EM has gotten to be wonderful in terms of structure quality, and these are around the 3 to 4 angstrom, so very high resolution for cryo-EM. We know what it looks like pre-fusion. Generally, the post-fusion state is much less well refined, but we have an idea, and you can see that they're really very different structures. This is showing you the resolution. It's pretty good for the core, but as you start to get out in these regions where antibodies bind and ACE2 bind and things like that, the resolution drops quite a bit, and we don't really have a good idea what it looks like. We're also missing all of the glycans on the surface. This is a glycoprotein, heavily glycosylated, and that's quite important, as I'll show you. Okay, so in my lab, if you are not familiar with my group, we do computer modeling, as Joel mentioned, and we do a lot of building of models. We can use the computer to build in models, given experimental data and things like that, bring different structures together. And then we also simulate motion. And so we use very large computers, and we try to track how these might move in time and come up with a model of what that motion looks like, and then understand from that motion, maybe there are opportunities to interfere. And so these are some of the models that we've built that correspond to the cartoons. This one on the left, this pre-fusion state is very similar to the cryo-EM, but it's got the full stalk going into the viral membrane. It has all the glycosylation that's been determined through things like mass spec. And so then we've also built models for some of these other states, like this fusion intermediate where it's attached to the host cell, and that is not known how that looks in the experiment. And then this final state here is the post-fusion where the viral and host cell membranes have been brought together. And so what we're trying to do now that we've built out these structures is understand how the virus gets from one to the next. And then one structure we still are working on building right now is this complex with ACE2, and we're nearly finished with that one. So this has led us to go after 2 different targets that we think could be useful. One, as our simulations have shown, these are the spike here on the left with all those receptor binding domains in the down state, and then occasionally one of them opens. And purple is the spot where the ACE2 is going to bind. And you can see in this down state where it spends most of its time that these blue glycans cover almost all of the surface. It's very difficult to get anything into the spike, including antibodies. And so this is heavily shielded through the glycosylation. Occasionally it does open up, and that's when it becomes susceptible to antibodies, but that's not very commonly seen. And in the structures for the wild type spike, it's not seen at all. There have to be mutations made to it to get it to open. So one possible target would be to counteract this ability of SARS-CoV-2 to hide. It actually hides much better than SARS did. And this is one of the reasons why it's thought that immune response is slow. Another one is that that S1 and S2 subunit that I showed in the cartoon, those need to detach from each other because the S1 is holding the S2 together and it won't undergo membrane fusion until the S1 leaves. That relied on host cell proteases in SARS and MERS and other coronaviruses. But in SARS-CoV-2, oops, sorry, you may have seen, I guess my animation's not working on that one. There's been a sequence insertion, an RRAR, a polybasic site, that's a substrate for host cell furin enzymes, and that detaches the S1 from the S2 before the viral particles egress from the host cell when they're being produced. And so that makes it much easier for that S1 subunit to detach. And so those are two things that are quite different from SARS. So on the first one, we've been spending a lot of time working on this. I wanted to show you a sample of one of our simulations and what we can look at. We wanted to know if we could keep the RBD from hiding. Is there a way that we can control that opening and closing? And so this is showing you our trimer of the spike in a surface, but we do this all in atomic detail with water and everything else. The green are the glycans. And so this animation is going to show you in our simulation that blue receptor binding domain moving up above the glycan shield where it can access an ACE2 and bind to it. And one of the things we noticed was that there's a spot that is not resolved in the experimental structures that's a transient pocket. And so it's very highly conserved. There's no mutations in this pocket in any known coronavirus, not even in SARS compared to SARS-CoV-2. So moving inside the spike, this is what that pocket looks like. There's a hole and there's a hydrophobic pocket with some hydrogen bonding opportunities. When we let the RBD close back down again, you can see that this entire pocket collapses and doesn't exist in that closed state. So if we could get a molecule to bind to that spot, it could be a target for an allosteric control of the spike. So working with Rob Rizzo's group at Stony Brook in applied math, we did that. And so these are just some little animations showing you. On the left is just that receptor binding domain and the domain that it's connected to underneath. And you can see that there's a hinge and that opens and closes and it largely stays closed. We can calculate free energy profiles and try to quantify how much it's up. This one we estimate is up about 1% of the time above the glycan shield. On the right side, this is when we have a small molecule dock there, it can't close with something bound to it. And so in this case, when we do those calculations, we show that it's 99% exposed, very difficult for the virus to evade the immune system. Another thing that we're working on is this shedding. I think this could be an Achilles heel to the virus. It has that furin site that lets it cut apart the S1 and the S2 without relying on the host cell surface enzymes. But that looks like it might be a weakness in the virus. And so that's this state down here where the S1 has to dissociate so the S2 can insert its fusion peptides in the host cell. This is known to be easier than in SARS, and it doesn't require ACE2 or any host cell proteases. It happens on its own, and there's some cryo-electron tomography images that have come out where they're able to develop a model where they actually show that some of these are in the post-fusion state already on the virus when it's produced. And so the question is, what triggers that dissociation and that transition? Because once it switches into that post-fusion state, it no longer has the ability to attach to a host cell and it's inactivated. So instead of a few of these on each virus, is there something we can do that will push it along and trigger all of them prematurely because then it can't get into host cells? And this is happening already. We just need to learn how it happens so we can accelerate it. So what do we need? Well, we're a computational lab, so clearly we need experiments. We just make predictions and need them to be tested. We've got some ideas of some initial molecules that we've identified through virtual screening, but they need to be optimized and we need some testing to see if they do shift the spike conformation. We need to know from structural data whether it's moved up and also whether having that RBD up more often leads to shedding of the S1, which we think that will happen because it weakens the interface. So we're hoping this could inactivate the virus. But we clearly need people that are experts on viruses. We need to know a lot more about the membrane composition as we're building these models. We need to know about the glycans, how they interact with the mucus on the surface. There's lots of things we don't know, but at least we're able to build some computer models that can help us pose some questions. I was asked to mention funding. We got this started, thankfully, from our OpenBPRC grant funding, and since then I've gotten a few other grants. We have one that I just submitted to NSF last week and are moving toward having an NIH grant, and it would be really good to have collaborators to get that done. And these are the people that did all the work in my lab. I've got a number of grad students working on this project, but also quite a few undergraduates. And it's nice because it's a system size that we've never simulated before and really is unprecedented in computational biology in general. These systems are getting into the millions of atoms for individual spikes. And then when we go out to the whole viral envelope, it's hundreds of millions to a billion atoms that we have to simulate. So it takes a big team to do it. And that's all I have. Thank you.